Hello everyone, welcome back to Dopamine Biology. Please make sure to leave a like and a comment. Do not forget to subscribe. Today, we will be talking about the main types of RNA. Of the many types of RNA, the three most well-known and most commonly studied are messenger RNA, ribosomal RNA, and transfer RNA, which are present in all organisms. These and other types of RNAs primarily carry out biochemical reactions similar to enzymes. As its name suggests, rRNA is a major component of ribosomes, comprising up to about 60% of the ribosome by mass, and providing the location where the messenger RNA binds. Ribosomes are composed of rRNA and proteins. The rRNA ensures the proper alignment of the messenger RNA, tRNA, and the ribosomes. The rRNA of the ribosome also has an enzymatic activity and catalyzes the formation of the peptide bond between two aligned amino acids during protein synthesis. As well as serving as genetic material, RNA has another critical function in virtually all organisms. It acts as a messenger, a short-lived intermediate communicating the information contained in our genes to the rest of the cell. We make RNA copies of our DNA genes with the help of RNA polymerase enzymes. RNA can act as a messenger in the process of ensuring genes are translated into proteins. If DNA serves as the complete library of cellular information, mRNA serves as a photocopy of specific information needed to serve as the instruction to make a protein. And the mRNA carries the message from the DNA, which controls all of the cellular activities in a cell. If a cell requires a certain protein to be synthesized, the gene for this product is turned on and the messenger RNA is synthesized. The messenger RNA then interacts with ribosomes and other cellular machinery to direct the synthesis of protein. It encodes by reading codons. The proteins go on to do jobs in the cell, and the unstable messenger RNAs simply are degraded. What happens after messenger RNA is made? You might wonder how a chain of sugars and bases, such as messenger RNA, can even serve as a template for forming a protein chain. It involves some clever adapter molecules. Amazingly, those adapters are also made up of RNA. They are called transfer RNAs or tRNAs. They use their bases to pair to their mirror images in the messenger RNA and line up the right amino acids to make the protein. tRNA carries the correct amino acid to the site of protein synthesis in the ribosome. It is the base pairing between the messenger RNA and tRNA that allows for the correct amino acid to be inserted in the polypeptide chain being synthesized. During protein synthesis, tRNA transfers specific amino acids corresponding to the messenger RNA sequence to the growing polypeptide chain in the ribosome. tRNA pairs with messenger RNA in a way in which each of its three nucleotides pairs with three nucleotides of messenger RNA. The set of the three nucleotides on the messenger RNA is known as a codon, while corresponding sequence on the tRNA is known as an anticodon. It is the base pairing between the tRNA and messenger RNA that allows for the correct amino acid to be inserted in the polypeptide chain being synthesized. Consequently, the messenger RNA is read according to the genetic code, which relates the DNA sequence to the amino acid sequence in proteins. Each group of three bases in messenger RNA constitutes a codon, and each codon specifies a particular amino acid. Each tRNA has a distinct anticodon triplet sequence that can form three complementary base pairs to one or more codons for an amino acid. The messenger RNA sequence is therefore used as a template to assemble the chain of amino acids that form a protein. To sum up, RNA is involved in the process of protein synthesis under the direction of DNA. Thanks for watching.